Rock Beisel, the chair of the department, and I want to welcome everyone to our annual Research Day Grand Rounds. It has been a particularly exciting development, and it is turning out to be quite an exciting day. So I want to welcome all faculty, trainees, members of other departments that have joined us. Just a couple of housekeeping and two thank yous. One, just reminding people to remain mute until we have questions and answers. Please, please put your questions in the chat as we go along, and that way we can take them in order, and then perhaps at the end we'll have people open up more. Also, I do invite people to please turn your cameras on if you feel comfortable. We've been at this for years now, looking at squares, and it's tough, but it's even tougher when you're looking at black squares with white names across the board. It's just, it's hard. I understand people are eating or distracted for other reasons, although I hope that we have everyone's full attention. So with that, two thank yous. The very first thank you absolutely goes to our Vice Chair of Research, Gloria Reeves. Gloria has done a Herculean job this year in pulling this together with her team and all of those that she engaged. But we have been isolated, separate, and disconnected for 2+ years. And Gloria had successfully, very successfully, set up a live in-person poster session with safety in mind, provided lunch that people could eat there. And there's those of you that are still, in the ballroom of the student center, able to watch together, but also all of us on the Zoom. Gloria, I cannot thank you enough for the work that you've put into this. It has been so far a great success, and looking forward to the rest of the day. The second thank you goes to all of our junior faculty and staff for the terrific posters that were put together and on display. And I hope that most of you had an opportunity to take a look at them. There has been an unusual number of very creative, diverse submissions that were on display, including staff from our psychiatric OT department to put up 2 posters, fascinating content, and led to discussion of, hey, let's collect some more data around this. So while it wasn't your classic poster, it was something that stimulated a lot of thought, a lot of discussion, and we're going to do some data collection for future work around those issues, all the way to lots of outstanding scientific posters. So thank you, but also thank you to the senior faculty that encouraged your junior folks to submit. And as you've heard me say before, it is the job of the senior faculty to groom and promote our junior faculty. And obviously we had 28 terrific posters, and behind that are the senior mentors that encourage faculty to present. So thank you. And with that, I want to hand over the podium, if you will, to our Vice Chair of Research, Dr. Gloria Reeves. Gloria?
Thank you so much, Jill. And I'd like to echo Jill's comments and praise. It's been a really fun morning and a really rewarding one for all of us. I wanted to first thank our research steering committee, our research leaders of the department, including Jill, and our co-presenter today, Dr. Todd Gould, for their strong support of letting us go a little rogue this year. So we have a tradition in the department of doing a trainee poster session once a year for this Department of Psychiatry Research Day. But we had the full support of our senior research leadership to offer a non-traditional poster option this year. So if things got derailed because of COVID and you had partial data, no data, you could still present your ideas. If you're a clinician and you don't have research skills or experience yet, you could work with the support of our research diversity subcommittee to develop a clinical QI project. So I think what it really helped us to do is we're shaking off the rust of the pandemic shutdown to really get together and critically discuss ideas, which is really what science is about. So we're really grateful for that opportunity. And as we had the poster session today, I was thinking so much about 2 years ago, sending out the email advising our researchers to shut down research per university protocol. It's been 2 years since that time. So it really speaks a lot to the resilience of the department to have such wonderful trainee, staff, and junior faculty posters. So congratulations to all. We do have a little contest, so we'll be doing an announcement at the end, the last few minutes of grand rounds, to give 2 awards related to the poster presentation. And I also wanted to formally acknowledge Catherine McDonald, my research assistant, who is the person who made sure there were posters, places for the posters to go, our COVID protocols, food, everything. She did a terrific job, in addition to presenting her own poster. So now I have the tremendous honor of giving an introduction to our 2 brilliant speakers today. I'm going to start with our guest speaker first, Dr. Carlos Zarate, who is the chief of the section of neurobiology and treatment of mood disorders and the chief of experimental therapeutics and pathophysiology at NIH. He obtained his medical degree at Catholic University of Cordoba in Argentina and did a clinical fellowship in psychopharmacology at McLean Hospital. To the great fortune of us all, in 2001, he joined the Mood and Anxiety Program at NIMH, and as I understand it, founded the Experimental Therapeutics and Pathophysiology branch. And as a humble researcher, it's hard to overstate the importance of that type of work that magnifies its impact, not just through the great research that's done, but also the training and the encouragement of young researchers to do high-level quality translational research. Dr. Zarate conducts neurobiological and proof of concept studies, looking at novel compounds and biomarkers. And we'd be here all day if I went through all of his honors, but I did want to mention that something good did happen in 2020. He was inducted into the National Academy of Medicine, which is a tremendous honor. So we're really, really grateful to have his expertise. And I would like to close his part of the bio by mentioning something that maybe doesn't get mentioned a lot, but he's quoted on the NIH Office of Equity, Diversity, and Inclusion in an interview advising Hispanic students why go to college. And at the end of that interview, he said something that I'm guessing, without knowing him personally, sums up his perspective and his approach to science. And he said, knowledge gained from college must combine with life and life experiences with the spirit of service to result in large benefits to society. So we're very grateful to have you come visit us, and we hope the next time it'll be in person. But we're delighted to learn from you today, Dr. Zarate. And then next, it's always fun to introduce someone you know very well. My colleague, Dr. Todd Gould, who's the Division Director of Translational and Basic Science Laboratories. He's a professor of psychiatry with joint appointments in Department of Pharmacology and Anatomy and Neurobiology. Dr. Gould completed his medical degree at University of Virginia. And as a child psychiatrist, I feel I must highlight that he completed a child research elective fellowship in the child psychiatry branch at NIMH, which, Todd, perhaps that's the reason you opted to start working with mice. I don't know that for a fact, but it might have been influential. Dr. Gould finished his fellowship training at NIMH in Lab of Molecular Pathophysiology and Experimental Therapeutics. Todd, as we all know, an expert in animal research with rodents to study mechanisms of rapid-acting antidepressant effect and development of novel treatments. On a personal level, I'd like to say I think we all in the research community certainly, and I think the rest of the faculty, think of Todd as an extremely attentive and thoughtful colleague. Todd is the one colleague I have who I've never caught him trying to read an email while I'm doing a Zoom call with him. He's always focused and really puts his effort and his thought, whether you're a trainee, a colleague, or a senior researcher. He has a very thoughtful approach to the conversations, which is an important building block of new ideas. I also like to say Todd is an outstanding teacher and mentor, and he serves the department in many roles, including his role on the mentorship committee. So that said, the presentation today could not be on a more timely and important topic, thinking about things like treatment refractory depression. But the reason that the presentation was selected was based on the discussion we had in the Research Steering Committee, that we wanted to talk about the importance of collaboration and the importance of bringing in different experts to solve very difficult problems. So it's really very exciting for us to hear about this collaboration among basic scientists, clinical researchers, clinicians, to address a very important public health need that's gained dramatic importance over the pandemic. So that said, I'm going to turn to Todd to organize how you guys are going to co-present. And we're, again, very honored to have both of you present at our return to the Department of Psychiatry Research Day tradition.
Great. Can everybody hear? Yes? Okay, good. And everybody can see the slides. So thank you so much for the kind introduction. And this is, I think, a nice story. There's a story that goes with this, and we're going to be focusing on the science, but a little bit more on the story. But this is an example, I think, of team neuroscience. And not one person could do this, but it was really a team and collaboration across many institutions. And, you know, we don't have enough time to give, you know, our thanks and gratitude to everybody who's pitched in this large project. But it's also an example of how we can get something from, you know, the bench to the clinic and from the clinic to the bench to come up with better treatments. And so I think that's a nice, and hopefully we can convey that in the story we present today. So the title of today's presentation is Translating the Acute and Sustained Mechanistic Effects of Ketamine into Safer and More Efficacious Rapid-Acting Antidepressants. This is my disclosure. I work for the Intramural Research Program at NIMH. And the objectives here are to understand the history of ketamine in the treatment of depression. Not too much in depth, but just give an overview, to become familiar with translational research on rapid-acting antidepressant agents using ketamine as a prototype. And then the third objective is to understand the pharmacology of promising therapeutics for treatment-resistant depression. I will start, then Todd will go in, and I'll wrap up the clinical aspects of this effort. Now, needless to say that treatment-resistant depression and suicide are challenging, and there's a tremendous need for improved therapeutics. If you can see towards, let me see if I can, pointer options. Here you can see that depression, there are adverse effects, including disruption to personal, social, occupational life. And there's also a tremendous risk for suicidal behavior. We do have many treatment options for depression, and they are largely grouped into three categories: psychosocial, where we have exercise, we have mindfulness, CBT, there's pharmacological, and there's neurostimulation that has taken off in the last several years, including ECT, RTMS, vagal nerve stimulations as example. This figure in the bottom depicts a major depressive episode, which lasts about 9 months without treatment. What you do with our standard treatments is you shorten the length of the major depressive episode. And the limitations of our current treatments are largely summarized here. About 30 to 40% fail these different options. There's low remission rates, and there's a considerable lag of onset of therapeutic effects. What do I mean by that? If you start treatment with a monoaminergic-based antidepressant, to get a full response, it takes probably 10, 14 weeks or longer. And that's with traditional monoaminergic-based antidepressants, as listed here. What next generation treatments will produce an antidepressant response within hours or a few days at most, and that is now possible, and will provide us a blueprint for future therapeutics that work rapidly, as does the prototype ketamine that we'll discuss. But there's an important need to identify the cellular and molecular targets, and that's where Todd comes in and other investigators. Now, this is a path of discovery for our current treatments for depression. We have over 36 different treatments, started with ipronizid or isoniazid. Early on, it was through accident serendipity, a drug that was given for tuberculosis, and some individuals developed what we say euphoria. They were in a backward medical unit wards where they were emaciated, they were not eating, they were failure to thrive, and so many difficult problems, lose weight. And what they notice is higher doses, they become euphoric. And And it occurred to somebody, well, maybe this could be used as an antidepressant. And other investigators went into the monoaminergic, serotonin, and norepinephrine and dopamine, and came up with a series of antidepressants. Largely are refinements of previous versions of our treatments. They're what we call Me Too or copycat drugs, but that has not resulted in greater efficacy, but more safety. In this study by the STAR-D, looking about 3,000 to 4,000 outpatients, 4 levels of care, all of the individuals starting with citalopram, the remission rates were about 36%, but it often took 6 months for half of the people to achieve remission. And you can see in the darker color here, purple, that by the end of 4 successive trials, still 1/3 do not achieve remission. So that speaks to the problem I mentioned, low remission rates and a considerable lag of onset of therapeutic effects. Now, when we came to NIMH in the year 2000, we decided, what do we pursue? Do we continue the monoaminergic-based pathway? We know what that led to, many treatments that help our patients, but we decided to go by the road of glutamate and neuroplasticity, especially with earlier data with glutamatergic modulators such as ketamine, an early proof of concept study that showed rapid antidepressant effects and had other properties. So we decided to study ketamine and other compounds. Ketamine is an NMDA antagonist. This is a cartoon where it blocks the PCP site within the channel. When it does so, it produces a series of side effects, dissociative side effects, physiological changes, and there's of course the risk of addiction. And this is a paper published last year that shows where ketamine binds within the at the PCP site. This is actually S-ketamine. And so one thing I highlight here is the short half-life of only a few hours, and I'll come back to that later on. So in our proof of concept study, we wanted to test whether blocking NMD receptors leads to a rapid antidepressant effect. Because of its side effects and abuse potential, and for ethical considerations, we studied individuals who had failed 6 or more antidepressants, 50% had previous suicide attempts, and many had failed ECT, a very effective treatment for depression. And this is a study where we tested a single dose in individuals who failed on average 6 antidepressants, Hamilton depression scores, up greater depression severity, down here minutes and hours. And what's remarkable of this, most of the trials before had been in weeks. You assess improvement within weeks. Here, we were looking at improvements within minutes. And you can see here within 2 hours, rapid antidepressant effects lasting a good part of the week. Towards the right are response rates with monoaminergic antidepressants in red. It's about 65% after 8 weeks. Taking a pill every day in individuals who do not have treatment-resistant depression. We see comparable response rates in individuals with treatment depression within a matter of hours. But you see that it peaks within 1 day and starts fading by the end of the week, but still significant by 7 days. And that led to a series of replication studies. Bipolar depression, you see the rapid onset within hours, lasting a good part of the week. And then another replication study, small studies, but very large effect size. And what you see is a very similar pattern of onset and offset of the antidepressant effects, which speaks to that we might be tapping into the neurobiology of the rapid response process. Then there have been a series of single and repeat infusion studies with racemic ketamine, that's R and S enantiomers of ketamine. And you can see here in this meta-analysis at day 1, the odds ratio is about 10 for responding to ketamine, and at week 1, it's 8. So very robust antidepressant effects. Not only that, earlier observations by our trainees and our fellows actually noticed that the suicidal thinking was improving rapidly. And that also has led to another path in the ketamine development. What we see here is a meta-analysis of individuals who had suicidal ideation in the context of their depression and who received a single infusion of ketamine in green compared to a control condition, midazolam or saline. And you can see about half of patients with suicidal ideation have a rapid improvement within 1 day, lasting a good part of the week. This is with racemic ketamine, R and S ketamine. And so this has huge implications in terms for clinicians because it provides opportunity to intervene very rapidly on the inpatient units, emergency room settings, and then hopefully get them safely to their doctor, to their family until the next treatment takes effect. Now, what has also been noticed is that ketamine also seems to have broad therapeutic effects different than our conventional antidepressants. And here I mentioned some of them: PTSD, OCD, social anxiety disorder, alcohol use disorders, cocaine dependence, pain, fatigue, anhedonia. And now the number, it's important to keep in mind that many of these studies are a few studies, they're small sample sizes and are in need of replication, but quite intriguing. It also seems to have improved biological indices of health. We see that evidence of neuroprotective properties, anti-inflammatory properties, bone markers are improved. Adipokines are also changed in a positive light, decrease in resistance, for example. So many of these are now being pursued in research. Now, the initial ketamine led to a series of studies by us and by others to try to test whether NMDA receptor antagonists with a more favorable side effect profile will lead to rapid antidepressant effects. And we say a number of studies by us and others with a broad NMDA antagonists, subunit selective NMDA antagonists really have not panned out. Here you see the channel, and you see the broad NMDA receptor antagonist, ketamine, S-ketamine. The only one that seems to have meet the 3 criteria: rapid onset, robust separation from placebo, and sustained action is racemic and S-ketamine. Memantine, Amenda for Alzheimer's, we did not find any effect. Lanisamine, there is some evidence of early antidepressant effects. Nitrous oxide, there is some positive evidence still being developed. But when you get into NR2B subunit selective antagonists, you do not see significant benefits. And even the glycine site modulators have not panned out for different reasons. So in our mind, we're fairly confident that NMDA receptor inhibition is not the primary mechanism of ketamine. Does not mean that it will not have antidepressant effects. It might have antidepressant effects, but it is not a ketamine-like drug. Now, that got us thinking. I mean, I'm here in the last couple of minutes of the first part of my talk. This was quite intriguing. We would see in our research patients this abrupt onset and fade away by the end of 1 week, beyond the half-life of ketamine, which is a few hours, and the dissociative side effects, which is only 40 minutes after the 40-minute infusion. So why is the antidepressant effect lasting so long? So that got us thinking. We measured a lot of things in our patients. One of them were metabolites. And so we wondered whether ketamine is actually a prodrug. And here we have racemic ketamine, that when you give it in patients, what you find here are within minutes, you have 24 metabolites or potentially 24 different drugs. Some of them may be side effects, some of them efficacy. But what we noticed here is a group of them, hydroxynorketamine metabolites, about 7 to 8, were hanging around for 3 to 7 days in our patients, and very coincidentally, similar to the efficacy component of ketamine. So we wondered whether these drugs, well, we went into a series of studies, which Todd will now take over and explain, and came up with a candidate drug, 2R6R HNK. And this is the HNK part of the story that Todd will summarize. I'm going to stop here, Todd, and you can take over. There you are.
All right, can everyone see my screen?
Yes.
Okay, so similar to Carlos, these are my disclosures, the most relevant of which is the last one. We do have patents and patent applications related to use of hydroxynorketamine for the treatment of depression. The patents are owned by the University of Maryland, or University of Maryland, Baltimore. And so as Carlos mentioned, part of what we're going to tell you is data and what we observed and where things are going, but just as important is the story about how things how things happened. And so in Carlos's last slide, last couple of slides, he mentioned a couple of things, right? The first was the clinical observation that other NMDA receptor antagonists have the same pharmacology at the NMDA receptor that ketamine does. They are not antidepressants. Okay? And the second was the observation that he made that ketamine metabolites are produced fairly rapidly and they're sustained for a bit of time after that ketamine infusion. And so Carlos and I, we've known each other for a while. Gloria mentioned that Carlos came to the NIMH in 2001 to run the experimental therapeutics branch at NIMH. I finished medical school in 2001 and started my postdoc at NIMH. So Carlos and I knew each other for the 6 years I was in training at NIMH, and then he stayed on, and I came to Maryland and established my own laboratory. But then we kept in touch and worked on a few projects together. And in 2014, we were at the Society for Biological Psychiatry meeting in New York City that year, and we were at one of those tables in the corner, and Carlos, we were sitting down. He said, "Todd, I made these observations. One, like I just said, the NMDA receptor antagonists don't recapitulate the antidepressant effects of ketamine, and I'm starting to think about maybe it's not ketamine itself, maybe it's the metabolites. And would you be willing to test some of the ketamine metabolites in your animal models of antidepressant efficacy?" And to be honest, my feeling at the time was that everyone knows that ketamine acts through NMDA receptor inhibition, but I respect Carlos, I respect his work, and maybe we'll have a nice negative study that comes out of our experiment. So that's how it started. And so shown on this slide is a bit more detailed view about the metabolism of ketamine. I'm not going to go through it in very much detail, other than to say that the intermediary metabolite is norketamine, and then there's a series of hydroxynorketamines that are formed, which are defined by the location. Let me just change my pointer. Of this hydroxyl group on this ring structure. And the most prevalent hydroxynorketamines, both in humans as well as animals that are produced after ketamine administration, are these 6 hydroxynorketamines. And the most prevalent 6 hydroxynorketamines are the 2R6R hydroxynorketamine and the 2S 6S hydroxynorketamine. 2R6R hydroxynorketamine is formed exclusively from R-ketamine, one of the 2 stereoisomers that make up racemic ketamine, and the 2S6S hydroxynorketamine is derived from S-ketamine. Also known at the time was that compared to ketamine, which has a high affinity for the NMDA receptor, the hydroxynorketamines, and in particular the 2R6R hydroxynorketamine, has very low affinity for the NMDA receptor, meaning that it binds less tightly to the NMDA receptor. And then one would predict that the NMDA-mediated side effects of ketamine, the dissociation, the dizziness, euphoria, the drug abuse potential, would not be observed if one were to administer the metabolites in the absence of giving ketamine. So I'm going to show you the results of some behavioral tests that are used in basic science laboratories to predict antidepressant efficacy. I could spend, maybe this is a talk for another time, I could spend an hour talking about the different behavioral tests and the rationales and their usefulness, and there are many limitations as well. But by and large, we aren't studying depression in mice, we're studying facets of depression, right? And in many cases, we're studying using tests as measures of antidepressant efficacy, and they may not be models of depression at all. But this is one test that's very commonly used in the preclinical literature. This is the forced swim test, where you place animals in cylinders of water and you measure how much time they struggle. And antidepressant drugs, shown here is the tricyclic dizipramine, if you give it 1 hour after injection, you have a robust decrease in immobility time. So the animals are struggling more, right? And ketamine, it does the same thing as dizipramine at 1 hour. And of course, that's not consistent with the time frame in which these drugs work clinically. However, where ketamine differs from dizipramine, where the effects last a couple hours, you have these sustained effects, again, as Carlos said, long after the drug is gone from the circulation, and in fact, long after the metabolites are gone as well. Another test I'll show you data from is the learned helplessness test. This test is performed in a 2-chamber shuttle box. On the first day of the test, mice are placed in one of the 2 chambers. They can't escape and they receive an electrical shock. On day 2 of the test, 24 hours later, we put the animals back in the same 2-chamber shuttle box. We give them the same electrical shock, and we ask whether, when given the opportunity to escape, do they try to escape? And it turns out that some animals will escape, and about 50% of the animals will become helpless. So even when given the opportunity to escape, they don't. They just sit there and receive the shock. And perhaps you could draw some parallels between the neurobiology, not this behavior per se, but the neurobiology underlying this behavior, and some of the symptoms of depression, such as helplessness. And then in this test, 24 hours later, we give a drug treatment and test again, and we test whether there is any reversal of this helpless behavior. So animals may choose to escape or continue to fail to escape. And so this is what happens when you give ketamine in this test. You see at doses of 10 and 30 milligrams per kilogram that you'll reverse that helpless behavior. And this is compared to fluoxetine and SSRI. With a single administration, you see no reversal of behavior, though I will note if you give fluoxetine for a couple of weeks, it's similar to its clinical time frame of efficacy, you do get reversal of helpless behavior in mice that receive fluoxetine. So there's a lot of data that I'm not going to show you, both in the because of time and to just be efficient to getting the point across. But then we moved forward and tested these 2 hydroxynorketamines. And I'll add that a critical component of this work was, at this point, a very young postdoc in my laboratory, Panos Sanos, who was in the laboratory leading all of these, certainly all of the behavioral pharmacology studies that I'll show you. And so we tested in different doses 2R6R hydroxynorketamine versus the 2S6S hydroxynorketamine. This is saline, a vehicle injection. This is ketamine. I already showed you it robustly reverses helpless behavior. And 2R6R hydroxynorketamine, beginning at a dose of 5 milligrams per kilogram, and then at a similar dose to ketamine, you see a similar reversal of helpless behavior. The 2S6S hydroxynorketamine also reverses helpless behavior, but note it takes about 75 milligrams per kilogram of the 2S6S hydroxynorketamine to have this ketamine-like effect. So we conclude that the 2R6R hydroxynorketamine is the more potent antidepressant-like drug in preclinical models. Now, I'm not going to show you all the different behavioral tests. One of the main focuses of my laboratory is behavioral pharmacology. So we have a number of behavioral tests that Panos performed in the laboratory. Includes the forced swim test that I showed you earlier, as well as chronic tests, such as chronic corticosteroid administration, as well as chronic defeat stress. In all these tests, we see that 2R6R hydroxynorketamine exerts both a rapid, robust, and a sustained antidepressant effect, very similar to that observed with ketamine. We also, and this is a study again emphasizing collaboration that included multiple other laboratories at the University of Maryland, Scott Thompson's laboratory in the Department of Physiology, and Edson Albuquerque, Edna Pierre's laboratory in the Department of Epidemiology. In fact, the co-first authors were a student in Eric, a student in Scott Thompson's laboratory, actually Edson Albuquerque's laboratory, and Tim, a PhD student in Scott Thompson's laboratory on this paper. And this is looking at functional effects of 2R6R hydroxynorketamine versus racemic ketamine on NMDA receptor function. And similar to the earlier binding studies, we found a very robust difference between the two. Ketamine has an IC50, where 50% of the response is inhibited with about 5 micromolar. And you're looking at a, I guess that's 40-fold, over 40-fold difference, over 200 micromolar for 2R6R H and K to 50% inhibit NMDA receptor responses. And so consistent with that, then we did tests that predict maybe some of the side effects of NMDA receptor antagonists like ketamine. One is just locomotion in an open field. Do mice change locomotor behavior? Ketamine clinically can result in euphoria, and we see some increase in locomotion and exploratory activity in mice. The 2R6R hydroxynorketamine, even up to a dose over 100 milligrams per kilogram, no change in distance traveled. One of the other side effects of ketamine clinically is dizziness, if not frank ataxia from the dose, even at subanesthetic doses. And this is a test where we're measuring a mouse's ability to hold on to a rotating drum. It's a test of dexterity and motor learning. Here you see the ketamine effect, robust change in behavior, 2R6R H and K up to over 100 milligrams per kilogram, no change in behavior at all, even in the early time points after administration. One of the main symptoms of side effects of ketamine is dissociation, out-of-body experiences, dissociation from sensory perception, changes in sensory perception. One way I think we can get at that is looking at pre-pulse inhibition of the startle response. Again, ketamine results in a robust deficit, and 2R6R H and K, now up to a dose nearly 400 milligrams per kilogram, has no changes in pre-pulse inhibition. Sometimes this is thought of as a measure of psychotomimetic actions of a drug. And again, there's no effects of 2R6R H and K on this measure at all. Another example of collaboration, this time within the department with Greg Elmer's laboratory at the NPRC. Greg's laboratory did all the work that's shown in this slide. So Greg assessed whether, compared to ketamine, there's an addiction potential of the 2R6R hydroxynorketamine. And so these are, in this case, these are mice that are hooked up to a syringe pump. Then when mice press the lever, they receive a small infusion of ketamine, right? And this is a IV self-administration procedure. Mice will readily press for administration of ketamine, and under the same conditions, they will not press for the 2R6R hydroxynorketamine. So again, predictive of low abuse potential of this molecule if given to humans. We've done a lot of work, and I'll just touch on some of the work related to 2R6R hydroxynorketamine's mechanism of action. The first work I'll show you was done in collaboration with Scott Thompson's laboratory. This is work by Lace Riggs, a PhD student in my laboratory who was co-mentored by Dr. Thompson throughout her dissertation. This is looking at AMPA receptor, another glutamate receptor, distinct from NMDA receptor, AMPA receptor mediated responses in a slice. And the bottom line here, I guess there's two things. Okay, one is that this measurement measures the strength of synaptic connections. And I think if you leave with anything, you leave with the knowledge that we think that these rapid-acting antidepressants are strengthening synaptic connections that are weakened in depression. So it rapidly does that. I don't show the data here, but actually when you withdraw the molecule, you can see a sustained synaptic potentiation. Our EC50 here is about 3 micromolar. We've also done whole cell recordings. This was done, again, part of Lace's paper, where this was done in collaboration. This experiment was done in collaboration with Edson Albuquerque and Edna Priera. We're measuring, rather than measuring on a extracellular level, field level, we're measuring individual responses in cells and looking at AMPA receptor mediated responses. And we have two outcomes. One is the frequency of those responses, and the other is the amplitude of those responses. Typically, when you see a change in the frequency of the responses, you think a presynaptic mechanism, more glutamate that comes into the synapse to activate AMPA receptors. When you see changes in the amplitude of the responses, you're thinking about the strength of individual receptor responses, which are a measurement of postsynaptic effects. Here we see a change in the frequency, but not the amplitude. It leads to a hypothesis that conclusion that hydroxynorketamine is inducing changes in presynaptic plasticity, which is actually a novel, relatively novel mechanism. We've done behavioral pharmacology to try to understand the role of the AMPA receptors. I know I think I'm moving a little fast because we're on a bit of a tight time schedule, so I apologize for that. But we're giving a drug that inhibits the AMPA glutamate receptor. And here we're giving the drug MBQX, which is a competitive antagonist of the AMPA receptor. And we're giving it right before we give the 2R6R-hydroxynorketamine, or also ketamine as a control. And we're testing the animals 24 hours later. And the bottom line is giving MBQX right before either ketamine or hydroxynorketamine blocks the antidepressant-like effects. We've used Western blots to measure AMPA receptors in the these are synaptoneurosome preps. So we're measuring AMPA receptors in the synapse of animals that received ketamine, either one or hydroxynorketamine 1 hour after administration or 24 hours after administration. Interestingly, we don't see changes in AMPA receptors at 1 hour, but we see an increase in AMPA receptors, both GLUA1 and GLUA2 subunits 24 hours after administration. So that then leads to a hypothesis that it's actually consistent with a presynaptic release mechanism early on, but then transitions to a postsynaptic mechanism that then maintains the response. And we asked that pharmacologically again, similar paradigm, giving drugs 23.5 hours later, then giving the AMPA receptor antagonist, and then 30 minutes later testing the animals in a behavioral assay. And again, we don't block in this case. I look at it as we reverse the antidepressant effects that have already been initiated during this earlier time frame, both with ketamine or the 2R6R-hydroxynorketamine. So this is I skipped over a lot of details, but this is our mechanistic hypothesis of how ketamine is working as an antidepressant. It's metabolized to 2R6R-hydroxynorketamine in the liver. H and K readily crosses the blood-brain barrier. It then acts to increase the probability of glutamate release. And this has been coupled with increase in high-frequency oscillations measured by EEG. We call this in the gamma range, 30 to 80 hertz oscillations. That induces a long-term potentiation-like event, resulting in a postsynaptic change, increase in AMPA receptors in the postsynaptic membrane. Ketamine's gone, 2R6R-hydroxynorketamine is gone, that presynaptic potentiation is gone. The long-term stability mechanism is this increase in AMPA receptors. That then segues. So Carlos is, when this is turned back to Carlos, he's going to tell you about a couple of things. One is he's going to tell you about the phase 1 and phase 2 planned phase 2 studies with the 2R6R-hydroxynorketamine for treatment-resistant depression. He'll also tell by this mechanistic framework about how hydroxynorketamine works, then suggests opportunities for alternative interventions. And one receptor that's known to work in a similar manner, regulating presynaptic release probability, is the mGluR2 receptor. It basically acts as an autoreceptor, and its activity dampens down release of glutamate. If you inhibit this receptor, for example, with an mGluR2 receptor antagonist, you do the same thing that 2R6R-hydroxynorketamine does. You increase probability of glutamate release. So it's an inhibition of an inhibition would lead to an increase in release. And just to, again, because of time, I'm just going to tell you our conclusions from a published study also led by Panos a couple years ago. And just reading that, we found that if we use an mGluR2 agonist prior to 2R6R-hydroxynorketamine, we prevent both the antidepressant-like effects of the 2R6R-hydroxynorketamine in multiple behavioral assays. And we also prevent the cortical EEG changes, that gamma power that may be related or necessary for the antidepressant effects of the molecule. We also showed that if we use knockout mice, mGluR2 knockout mice, they don't show antidepressant-like effects of the 2R6R-hydroxynorketamine, and they don't show the gamma power increases as well. And finally, we found synergistic effects between a sub-effective dose of the 2R6R hydroxy norketamine that itself does not exude antidepressant-like effects with an mGluR2/3 selective antagonist. Both on their own, if the dose is given, don't have antidepressant-like effects. They have a synergistic effects in combination. Again, suggestive of a convergence of mechanism between hydroxynorketamine and the mGluR2 receptor. And again, in gamma power as a translational biomarker that I think Carlos will tell you about, that we look at in humans as a marker of potential marker and also maybe part of the mechanism of these drugs. And then finally, again, quickly, Carlos and I engaged a couple of years ago, and full credit to Carlos. This is again, his idea. We've had for years data with MBQX in animal models that it blocks and also reverses the antidepressant effects of ketamine. I showed you that earlier. And Carlos wanted to ask this question in humans. And MBQX is not a human approved drug, but there is a drug, perampanel. It's an anticonvulsant. It also blocks the AMPA receptor. It does it in a different manner. It's non-competitive versus MBQX is competitive. But we wrote this NIH bench to bedside grant proposal. Initially, work would be done in my laboratory, asking, does perampanel do the same thing as MBQX, even though it has a different mechanism at the receptor? Is there correlation between changes in gamma power? Does perampanel block the gamma power changes? If the answer was positive, this would move on to what's going on now, a phase 2 study, proof of concept, experimental therapeutics trial in Carlos's branch at NIMH. And again, because of time, I'll just tell you the bottom line is that perampanel, similar to MBQX, does block the antidepressant effects of ketamine on multiple measures, including the forced swim test, as well as chronic multimodal stress paradigm. And it also blocks, this is when you give ketamine, this is this increase in gamma power, look at the 40 to actually in high gamma, 140 hertz range, you see this increase in activity, and that's completely blocked by perampanel pre-administration. This then allows us to move forward in clinical studies where we can do actually a very similar experiment, which Carlos will tell you about. That's it on my end.
Okay, so go back to slides here. All right, where are we? Part 2. All right, can everybody see part 2? All right. Okay, so this is an ongoing work in collaboration with Todd. So we have two groups. One is mechanistic studies into the rapid onset, which is AMPA signaling in the antidepressant effects of ketamine. The second is investigating novel targets. So you could see where the path has led us to, mGluR2, why not study a drug, mGluR2 antagonist in humans, or 2/3 antagonist, and then HNK, hydroxynorketamine in humans, and that's where we are. Here, just to recapitulate, you have ketamine, HNK produces this glutamate burst, it activates AMPA receptors, and so the question here, if AMPA throughput or activity is important, can we block it? Todd has elegantly showed that in preclinical studies you can do with MBQX, but what about in humans? And so the question here that we propose, an example of back and forth bench to bedside, is AMPA signaling critical to racemic ketamine's acute and continued antidepressant effects. And if you recall that Todd showed that with pre-treatment with perampanel or MBQX, you can block the acute antidepressant effects of ketamine and HNK, ketamine, I'm sorry, and that those that achieve behavioral effects, acute response, can you reverse it at 23 hours after the onset of the antidepressant effects. And so here, perampanel is an AMPA antagonist. It's an AMPA antagonist, anti-epileptic drug. And Todd already showed it that it blocks in several different models, the forced swim test. But the problem we have, at least in translation, is a translational biomarker. How do we know that we are at the intended target? And that's referred as target engagement. In work we didn't have time to show, towards the bottom right is What is well known is that if you give ketamine, you produce increases in gamma power, which in part is due to AMP activation. And Todd already talked about that. In this earlier study, he showed that with both ketamine and hydroxynorketamine, H&K, you get increases of gamma power, and you can block that. So the question we asked, can we do the same in humans? And here is the design of the study. So subjects will get open label treatment with ketamine, and they would be pre-treated with perampanel or placebo. Already Todd mentioned that in animals or rodents, you can block the acute effects. But some of these will go on, those who receive placebo, to have a continued antidepressant effect. And so at a second stage, subjects would also receive a day or so later perampanel to see if we can block the continued antidepressant effects of ketamine. That study is ongoing right now. But in addition, we get a series of biomarkers, including resting state. We get MRS to see if we can measure the glutamate burst in real time. And then we obtain MEG magnetic encephalography, where you can measure gamma power. And we have already measured that in our patients treated with ketamine. And so this is an example of this is the bench to bedside award that's going on, a collaboration in both institutes. Todd also showed that one possibility of how ketamine and hydroxynorketamine might work is at mGluR2 receptors, where if you inhibit mGluR2, you have the so-called glutamate burst, AMP activation, and so on and so forth. He already showed that a single infusion in rodents can increase extracellular levels of glutamate, curiously around the same time frame that occurs in humans. This is with mGluR2 agonist, which is 268. He already showed you in the Panozanos paper that was published several different evidence that suggest that mGluR2 is important in the mechanism of ketamine in its presynaptic release. Now, we had looked for a company high and low about who might have an mGluR2/3 antagonist, and we found a collaborator, which is Taisho. A series of experiments were done preclinically, which I collaborated with them, and also clinically. And what you find with one drug, which is called TS161, which is really 894, which happens to be a prodrug. Because of problems of absorption, we give another compound, 292, that eventually converts to another form called 894. And when you look at those compounds, what you find is it has affinity for the human mGlu2 and GLU3 receptors. You can see the antagonistic activity is quite comparable to rodents. So it does offer a nice potential target and translational biomarker. Now, a series of other experiments went on to characterize its pharmacological, pharmacodynamic effects. And what you see here is after the drug 292 converts to 894, you see that it achieves in cerebrospinal fluid a concentration of 10 nanograms ml, which is precisely where you will get the antagonist activity. So we predict that this would have antidepressant activity at the dose of 100 milligrams a day. So this is the study that is now ongoing. We completed a phase 1 during the pandemic, and right now on the research unit, we're doing a treatment-resistant depression study. Subjects receive 50, 100 milligrams of TS161 for 3 weeks. It's oral, and then they cross over to the other condition. In addition, we get we get series of biomarkers, including peripheral measures and central measures such as bold response, fMRI, several times during the administration of drug or placebo. And we get MEG gamma power, so we can see if it's a potentially translatable to go back to the bench and to work out. So that's the study that's ongoing right now. And let's see, I think we have, sorry. And then, sorry, maybe I skipped a slide. And then this is the hydroxynorketamine study, which is pretty much the same design. Todd presented evidence that its antidepressant effects are not NMDA receptor due to an NMDA receptor inhibition, produces series AMP activation, series of intracellular signaling cascade changes consistent with ketamine, does not have the abuse side effect potential of ketamine. It does in preclinical studies show increases in gamma power. And we have, this represents really a multi-institutional effort. Craig Thomas at the National Center for Advanced Translational Science, NCATS, also includes Ruin Model at the National Institute of Aging. Folks from NIDA are involved as well, because we're pursuing treatment-resistant depression. We have recently completed a single ascending dose of phase 1. Multiple ascending dose should start soon, and we anticipate testing hydroxynorketamine on a research unit early next year. And
Dr.
we'll also,
Zarate, I'm sorry to interrupt you, but you've already galvanized a lot of questions in the chat. I wanted to make sure we had
okay.
a chance to get to a couple
yes,
of them.
and
but
so that was
got a
my
lot of
last
interest.
slide. Okay.
So thank you so much for a really fascinating talk and a true spirit to collaboration and the importance of collaborative research to answer difficult questions. So it looks like there are, so we have time for a few questions. It looks like some are more related to the research design or the data, and then one question about the clinical. So Todd, I don't know if you see those, if you want to triage that.
Yeah, so I can, Mitchie Kelly in the Department of Anatomy and Neurology had a couple of questions. One was about regarding AMPA receptor PAMs, ampakines, whether they have an antidepressant like profile, and preclinically they do. There's positive data. And then there's at least one molecule that's in phase 2 or 3, I think 3 development, right? Carlos, the Takeda compound, the AMPA receptor PAM for treatment-resistant depression.
Yes, I think it's TAX 653 by Neurocrine is being developed also for cognition as well. Historically, not all of them increase BDNF, which is believed to be important, and there's some, I think, suggestion that there might be tachyphylaxis that effects wear off fairly quickly. And then there's the issue of seizures at higher doses in theory. I think that's all because of the time remaining. The other is, what other?
I could address briefly Dr. Kelly's question about the U-shaped response.
Yes.
Which he picked up from the preclinical data with both learned helplessness as well as forced swim tests. We also see that in other behavioral assays as well. We don't think it's a side effect of ketamine related to locomotion per se. We do think it's related to ketamine's action to the NMDA receptor. So one of the basic principles of synaptic potentiation, and I'll, since Scott Thompson is on the line here, I'll just throw out everything I've learned about synaptic physiology I get from Scott and Edson and Edna and students in the laboratory as well. But the NMDA receptor activity is necessary for potentiation of excitatory synapses. And our thinking is that at these higher doses of ketamine, that we're effectively inhibiting the NMDA receptor and preventing synaptic potentiation. We have a lot of behavioral data supporting that, and we have now Kyle Brown, who's a relatively new postdoc in the laboratory, now has EPHIS data support this as the mechanism related to the U-shaped dose response curve, and we're putting a paper together that's going to probably be submitted pretty soon, somewhat describing our thoughts on that phenomenon.
It looks like there's also a question from one of our senior clinicians, perhaps Dr. Zarate could respond to, about if recreational use of ketamine might contribute to chronic depression. So there's a description of a patient in their mid-30s who had recurring severe depression, suicidality, prior treatment with ECT, recent ECT moderately effective. So he was asking about use of ketamine recreationally or abuse of ketamine, if that it could contribute to risk of depression.
So could it increase the risk of depression? Well, first of all, depression and addiction are in substance use disorders are commonly comorbid, 30, 40, 50%, even higher for bipolar disorder. So What's not clear is there have been studies, is it the chicken or the egg? Do you start with substance that abuse problems and then it leads to depression, or do you start with depression and leads to substance abuse? And you can find, patients either way. And others have proposed, well, maybe they're self-treating themselves. Clearly, you'll hear stories of people who say, I took ketamine and my depression went away. I was, and I was taking cocaine and it went away. But again, that's, not really systematic. But I do say there is a lot of overlap of that. And in some cases, it's possible it might contribute to depression, and in other cases not. The most important thing is that we do know that the comorbid use of substances with depression will increase the chronicity of depression, will increase the relapses, and other medical consequences. And so it's not going to help long term. And so that's why ketamine or Spravato is given under the medical supervision. You have to be trained for it, and it's only given in the doctor's office, and it's monitored closely.
Just going to follow up on the second part of that question. Would it be more likely then that the person would be refractory to ketamine treatment now if he used it recreationally when he was younger?
We don't have evidence that's the case. We have had several hundred patients treated over the years with ketamine in our studies, and we did not exclude past substance use disorder. We looked at that; it didn't seem to diminish the antidepressant effects or enhance the probability. We do know that there's some evidence that if you have a family history of alcoholism, that might increase the probability of responding to ketamine. And keep in mind that alcohol is an NMDA antagonist in certain ways. Now, that doesn't mean you should, only give it to family members, what I mean, right?
Mm-hmm. So I know that there are some more questions on there. I just wanted to pause for a minute and announce the recipient of the awards, because our clinical trainees have to head back. And then I invite folks who want to stay on to continue with some questions, if that's okay, Dr. Zarate, for
Absolutely.
another 5 minutes. But I wanted to announce, so first of all, thank you to Dr. Sarah Clark, who organized the research poster judging session again, and our judges, Dr. Sonia Bansal, Stephanie Hare, Sean Davis, Samantha Reeves, Shimendi Savaratne, Greg Elmer, and Alicia Luxted. So thank you so much. And the award, there's 2 awards. One is for a traditional research poster, and one is a non-traditional. And the traditional poster award is $500 reimbursement towards conference fees or purchase of something educational, textbook, that sort of thing. And then for the non-traditional poster, the award is a $250 reimbursement. So I'll reach out to you, give you the details of that. But our winner for the traditional poster, the title is Characterizing the Relationship Between Flexibility, PTSD, Symptom Severity, and Implementation of an In-the-Moment Mobile-Based Intervention in Women with Substance Use Disorders. So Dr. Cohen, perhaps somebody's working on some of these questions that you're interested in. So the winner is Sarah Uhouse for the traditional poster. So congratulations, Sarah. Hopefully I said your last name correctly. The second award for the non-traditional poster, the title is Statewide Usage of Apparent Web-Based Training for Adolescent Substance Use, Preliminary Findings. And the awardee is Chantel Smith. So a hearty congratulations, and thank you so much to the hard work of all the trainees. And I suspect we should have scheduled a lot longer, because this is a really interesting topic and a lot of questions. So those who can stay on, I invite you to stay on the next 10 minutes, maybe, to finish up with some questions. But before people disperse, I wanted to thank so much our speakers who did an amazing job in not just conveying the science, but the collaboration and the work that led to those important findings. So first, a big round of applause. Thank you. All right. So Todd, I don't know where we left off. I think there was a question from Jim Walls. Is gamma power a general measure of neuronal activation, or something more specific like attention or cognitive finding?
Yeah, that's a good question from Jim. I guess another also changes in gamma power were previously thought to be related to the psychotomimetic effects of ketamine as well. And so I think it's non-specific, right? It's a measure of high frequency synchronized neuronal activity. And so it certainly could be used in measurement in cognitive or attentional tasks. What we're interested in, when I say we, I'm thinking about myself, and also, mentions Dr. Thompson, who's very active in these collaborations and thinking about how these drugs may be acting. That these high frequency synchronized activities have a parallel to what's sufficient to induce potentiation of synapses in slices. So the traditional mechanism to potentiate excitatory synapse is to give repeated high frequency stimulation. And so our hypothesis is that change in gamma power, in relatively transient change, an increase in gamma power, functions as a stimulation to the synapse, then combined with increased probability of glutamate release, leads to a postsynaptic potentiation, a LTP-like phenomenon in vivo. Carlos, do you have anything?
Yeah, and I think in humans, you find you find changes with ketamine up to 6 to 9 hours after the infusion. It's consistent with, or it is cortical excitability. Could be, as Todd mentioned, a putative marker of synaptic potentiation. You even can find some data suggests you can find it very acutely with the start of the ketamine infusion. But we know that you can see gamma power changes involved, in visual cortex and the auditory cortex, and but you can induce plasticity or gamma power changes with specific tasks, a visual task, an auditory task. And what we do find when we find that the gamma-induced increases with, for example, somatosensory task, it correlates with antidepressant efficacy. So we see with ketamine, I'm sorry, antidepressant efficacy. So it does, it's a potential, marker of plasticity, but we don't know the precise mechanisms. And when it's general, we know it's parvalbumin, and it's there, and there's these the ping circuits and the ing circuit microcircuits, but we just don't really know. But we are using it as a cross-species biomarker for target engagement. We don't know if it's a dose-dependent effect as well. There was something else about.
Looks like there was a question from Mark Cavarda. Mark, are you on? Did you want to mute and ask your question?
Sure. I mean, it's exactly as I typed it. But basically, how do you think about 2S, 6S, or S-ketamine? The approved version of Spravato was, I guess, efficacious enough to be approved, but at 1 milligram per kilogram or 0.5 milligrams per kilogram, and not 10 and 20 times the dose that you might anticipate based on the potency studies from Todd and Panos's work. So should we not be bothering with S-ketamine? What's your take on it?
Thank you, Mark. That was my question as well. So I appreciate your asking that.
So the question is why we're not giving higher doses, or what?
Well, I mean, if S-ketamine is 10 to 20 times less potent than 2R, 6R, why don't we need a much bigger dose? How did it get approved, and why is that the one that we have available in clinic easily?
Well, I think that's assuming it's the NMDA receptor, right? I think what you're getting at. We're arguing that maybe part of it is, but maybe not. And as Todd already showed, you also have 2S6S has antidepressant-like properties. We think the mechanism is something different than NMDA receptor inhibition. I would say there has been some studies looking at lower doses of racemic ketamine. I wouldn't, even though it was 0.1, 0.2, 0.5, and I think 1 milligram a kilogram, it wasn't really a dose finding study, but you find the optimal dose with racemic is 0.5, as all the studies have been done. Even in the studies way back in schizophrenia, it's always been 0.5. But at higher dose, you didn't seem to get a better antidepressant effect, and you got more dissociation. So another possibility is you always wonder why companies or industry chooses the drugs they do. And, they try to find where you have the therapeutic effect, but not running into much side effects. So it's possible as you give higher dose, very likely as you give higher dose of S-ketamine, you start, running into more dissociation and and blood pressure problems. And so that would not be well tolerated and wouldn't, you would have to convince the FDA that there's some benefit. So we just don't know. There's very little in dose finding for racemic, some for S-ketamine, but not a whole lot. And, I don't know, Todd, do you have any other?
And maybe a couple of things to add. But one is that so ketamine, racemic ketamine, has the two stereoisomers, the S-ketamine and the R-ketamine, and Janssen decided to develop the S-ketamine, which is now approved for intranasal administration. The preclinical data, both from my laboratory as well as other laboratories, actually indicates the R-ketamine. The R stereoisomer is actually the most potent antidepressant compared to S-ketamine. But like Carlos said, we do find that the 2S6S hydroxynorketamine is antidepressant. It's simply not as potent as the 2R 6R hydroxynorketamine. There's also a metabolite that I showed you in the slide that's intermediate between ketamine and hydroxynorketamine. That's the norketamines. And we haven't studied the norketamines in my laboratory, but Kenji Hashimoto's group in China has, and he's actually found that the S norketamine is a fairly potent antidepressant. So it may be that depending on the metabolite, you may find that the S versus the R may be you are finding the more potent antidepressant. And that unknown target that's different from the NMDA receptor that's responsive for these the antidepressant activity, these molecules, it may be that they don't completely track with the S stereoisomer versus the R stereoisomer. So it is possible that, for example, that S-ketamine is metabolized to S-norketamine, and that may be the more active molecule. And the bottom line for all of these is we need to wait. Right? We can study. I say that around the world, thousands of laboratories have cured depression in mice, and my laboratory has cured depression in mice many dozens of times, and it doesn't really matter that much. That's not the end game. So what really matters is what we see in people. We'll know hopefully pretty soon, maybe starting next year with the H&K phase 2b study that Carlos will be initiating. R-ketamine, okay, the R stereoisomer of RS-ketamine is in clinical development. I think that's in phase 3 or maybe phase 2 now, between phase 2 and phase 3. So and there have been case reports from case series more from South America suggesting antidepressant effects of the R-ketamine, but we'll wait and see what happens. But I think there's a reason for excitement, and that our understanding, being able to answer some of these questions, Mark, that you asked, hopefully many of them will be answered in the next couple of years.
Yeah, I think that leads us to our final question. I think Scott Thompson is looking for a preview of the human data. If you have any teasers you want to throw out.
That's up to Todd to say.
I thought it was up to you to say, Carlos.
Well, I don't know. Do we have any permission to say or not?
We can talk about the single ascending dose. So we have a dose from 0.1 milligrams per kilogram to 4 milligrams per kilogram. I'd say that nothing has been unexpected to this point, and we have the clear go ahead to move forward with the multiple ascending dose study.
So you don't want to say anything whether you see dissociative side effects with higher doses or gamma power changes?
But I said that, didn't I say the results are as expected, or?
He knows this is being recorded, so he's keeping it close to the vest. All right. Well, thank you so much. This has been a really stimulating discussion, and really wonderful example of again, the collaboration. So we're going to end this formal part of the research day. And then I know Todd is organizing a subgroup of us to meet, perhaps to give a few minutes break, kind of give people a few minutes in between, and then we'll log in for that other separate meeting. So thank you so much, everybody.
Thank you. Bye. There's another line, right?